Our next speaker is Dr. Anthony Windebank. He is a professor of neurology at the Mayo Clinic in Rochester. He earned his BA and MA degrees in biochemistry and his follow-on degree at the University of Oxford in England, followed by postdoctoral training at the University of Oxford College of Medicine, Mayo Clinic, and Washington University in St. Louis. Dr. Windebank served as the Dean of the Mayo Graduate School from 1992 to 1998, and the Dean of the Mayo Medical School from 1998 to 2005. Since 2005, he's been involved in the development and leadership of the Center for Translational Science Activities and the Center for Regenerative Medicine at the Mayo Clinic. He's also a neurologist who specializes in the diagnosis and treatment of patients with diseases of the peripheral nervous system and spinal cord. Dr. Windebank has been involved in designing or conducting more than 50 clinical trials and clinical studies since 1980. With that, I would like to welcome Dr. Windebank.
Thanks very much. So, part of my role here today is to talk about someone who has been on the receiving side of funding from Department of Defense mechanisms. And this is something that got involved with about 7 or 8 years ago with the initiation of the AFIRM funding mechanism. And what I want to do is to talk about the very positive aspects of that funding and how it can drive translational research forward. And then also what are some of the different ways that as an investigator you're involved with that funding compared with traditional NIH funding. So I've been fortunate to have NIH grants for about 35 years now, and I'm very familiar with that mechanism, but have been excited and challenged working within the Department of Defense structure. But the bottom line is that this is a very, very positive way if you are interested in moving translation forward. So just briefly to introduce part of the team and to follow up on the Connect, Collaborate, and Cure. One of the things that I'll talk about as well is that funding drives the direction of your research. My research team, the team I'm associated with, has a lot of different aspects of activities within the regenerative medicine field. And I'm going to be talking mainly about the Department of Defense funded work in peripheral nerve repair and how that can be accelerated or slowed down according to funding, because when you have a large group, your priorities are in part judged by, part directed by what you can do, where's the technology, but where's the funding going. So I'm going to talk mainly about peripheral nerve repair over the next 5-10 minutes, and also to stress one of the first parts about the AFIRM funding mechanism, which was to drive collaboration. So I've been involved in nerve repair and nerve regeneration, and because of the AFIRM collaboration, we're really able to set up very productive collaborations with investigators at the Cleveland Clinic, Rutgers, and MIT in terms of developing new tissue engineering strategies to drive towards nerve repair. And it also really helps cement my collaboration, which has been going on for several years with my colleague Dr. Mike Yazemski, who will be talking in a couple of minutes, in how to bring together technologies from different fields to bear on this problem. So briefly, the points I want to make is that the Department of Defense funding can really drive collaboration. And I think to the extent that it can do that, both between academic investigators and between military and academic investigators, and then ultimately between the investigators and the commercial interests that are necessary for, ultimately for translation. It can drive the direction of research. It can also drive the speed and completion of research. And I'll make one of the points here about the difference between NIH funding and Department of Defense funding is that as an NIH-funded investigator, you put a great deal of effort into writing that R01 proposal, for example, and sending it to Washington, and if you're very fortunate, it comes back with a good enough priority score to be funded. And basically the message, as all of you know who have NIH funding, is here's the money, do what you're planning to do, this looks really exciting, do your best, and when it comes to renewal in 5 years' time, tell us how successful you've been. It's not quite that open, but it's very much an investigator-driven process where you have kind of almost complete freedom to operate and change direction of your research. The process with the Department of Defense is different because it's essentially, most of the mechanisms are contract-based. You contract to carry out research with the idea of delivering a product. And in order to deliver that product, you work carefully with the agencies to assure that you're making the milestones that you've set out that you need to get to the goal. Now, when it started, that seemed to be maybe a bit of an onerous process, but in fact, reflecting on it, it becomes a very good way of keeping the research, I'll say, razor focused on your translational objective, which is incredibly important. I was recently invited to write a commentary for Science Translational Medicine about how to increase the speed of translation. And one of the examples that I used was in fact AFIRM, and that this mechanism of promoting collaboration and of having milestone-driven research is a very effective way to move things forward more quickly. But as with all things, you're highly dependent on adequate and continued funding, and I'll come back a little bit about that at the end. Oops, sorry. So nerve injury is a major problem. We've heard about the goals of DoD programs are to return wounded warriors, to restore form and function, if possible to return them to duty or back to a good, productive life. And nerve injuries are one of the major causes of ongoing permanent disability. And just to give one example, my colleague Dr. Yazemski took this picture in one of the forward stations of an arm injury from an IED. And I'm just using this to give the example that this wounded warrior's hand is essentially completely normal, but because so many of the structures in the forearm have been severed, that hand is going to have to be amputated and will be amputated. And the major structure that can't be repaired here, and which is essential to hand function, are the peripheral nerves. In order to animate the hand, to feel it, that it's not a painful hand, you have to restore the nerve function. Everything's important, but the nerve function is critical. Now, one of the things that we've thought a lot about is how can we accelerate this process? And without going through a lot of details, it's very complicated to get from a concept in the lab, say a new tissue engineering biomaterial product, and then work that all the way through to patient care. And so thinking throughout this process of how can you accelerate the process of translation? Other ways that you can kind of move through things faster without having to go through all the steps in a linear fashion. And this is one of the things that we've been working towards with the nerve injury program. So one of the things was to develop a standardized small animal model for nerve repair that multiple investigators would agree would be useful for testing out new products. So, I gave the example of the 6 different investigators working in the area who came together, came with different products, different constructs, and said, now we'll put these through the same model. And put them through the same animal model. Now, I'll just give an example of why this is important. So, a couple of years ago, 2 years ago now, we published a systematic review in the journal Biomaterials of animal models used to study nerve regeneration in tissue-engineered scaffolds, so very, very focused. And we identified that there were 416 papers or reports over a period of 50 years with 70 synthetic materials, 8 species, 17 different nerves had been used. The gaps ranged from 1 millimeter to 90 millimeters, and there were 20 different assessment techniques. Now, what that meant was that it's virtually impossible to look at one paper and say there's this material, and then look at another paper with another material and figure out, well, which one's better? Because they will have used probably different nerves, different assessment materials that make that very, very, very different. So one of the major things that we were able to collaborate on was here's a small animal model that we all agree on. And so this was a standardized rat 1 centimeter sciatic nerve model. And for example, here there are half a dozen or 8 different materials that were used and directly compared in this model. And then when the best material was identified by this process, that was then validated in a second investigator's lab. So that's an important process as well. It's something that NIH has embraced in terms of important translational discoveries is the importance of validation. So one thing can be done in one lab, you figure out, okay, that's the best. Now, can you take that and put it in the hands of other investigators and validate it? And the material, or rather the types of assessments used to evaluate the outcome here were functional ones, physiological ones, and anatomical ones. And I'm not going to go through lots and lots of data details here because that's not really important for today's discussion. But one material was identified, polycaprolactone fumarate, which seemed to support nerve regeneration best with minimal host tissue reaction and to produce very normal looking regenerated nerve. So then the next step in the process is how to move that, instead of going through small animals and then up to a large animal and eventually up to a human trial, is there a way to do your safety trial and move it very quickly into humans? And if you were at a talk I gave earlier this morning about amyotrophic lateral sclerosis, it's parallel to that, where we went straight from a rodent model, or in that case a rabbit model, into humans, skipping over the large animals by choosing an appropriate safe human model. And in this case, what we're doing is using the defect that's left after a sural nerve biopsy. So it's a very standard diagnostic procedure. 6 centimeters of nerve is removed. And in the clinical trial that's under review at the FDA at the moment, it's to repair that nerve defect, which is normally not repaired at all, with a PCLF scaffold, so a new novel biomaterial. Then the next step in how to try and move things forward faster is that all of us that work in nerve repair believe that the ultimate nerve repair device is going to be a composite. It's not going to be a biomaterial, it's not just going to be cells. It needs structure, and it will need a biological component, which will be at least cells. So it's going to be a combination device. In addition, if we can then take the pieces of the combination through individual trials and then put them together, that makes the regulatory pathway much more straightforward. So we have very soon to begin a safety trial with the new biomaterial that will be done in the sural nerve biopsy. We're also collaborating then with colleagues at the Miami Project to Cure Paralysis, who have developed Schwann cells and have that approved through the IND to treat spinal cord injury, and are treating patients with spinal cord injury. By sharing their protocols, we'll then have a cell-based therapy that we can combine with our biomaterial that the FDA has already seen it in the nervous system, and we hope we'll be comfortable moving this forward into nerve repair. So additional strategies that we've been using to try and accelerate this nerve repair is to do something that's, I think, unique, which is to actually have a biomaterials-based GMP facility within an academic institution. So this is something we're setting up at the moment, where we will actually be able to construct first-in-human biomaterial-based constructs that can go into clinical trials. So not having to license this out to a commercial partner, but able to do those first-in-human trials ourselves. And that was in part driven by the fact that it was very difficult to find an appropriate commercial partner who was interested in this. And then when we found one, it actually turned into something of a semi-disaster. So it was, if we're in control of the process, if we can control the construction, the fabrication of the materials that we're going to put into our patients, we'll be very, very much more comfortable about it. The other is to devise or create a new role within our research team, which is something we call the translational integrator. So now when you're working in the space where you're making a combination device with cells and materials, it's going through GMP, it's going through FDA, it's going through the FDA, it's being funded by, in this case, predominantly the Department of Defense, and eventually you're going to want to commercialize it. Somebody who can keep all of those things running and moving together is an incredibly important function. We've identified that and modeled that and really believe it's a key success in accelerating translation. So I'll just finish up by saying here's some of the things that in retrospect and in thinking about I would identify as accelerators or delayers. So my colleague Mike was saying at the beginning, the biggest word in research is the big F, which is funding. So the accelerators driving this forward was clearly the funding through AFIRM 1. I'll come back as I get into the delayers and say that because of the funding structure and the less amount of funding available through AFIRM 2, you can see that slowing down the process, because within that big laboratory setting, it means the number of people that can work on these proposals become less. I've really become a strong believer in the contract milestone-based process that allows you to move things forward translationally quickly, which is modeled very well with the DoD. The huge acceleration you get from collaboration. The team collaboration moves things forward faster, period. There's also, some of you may be aware of, something that's working to our advantage here in the FDA, which is a new process and policy that they introduced almost exactly a year ago called early feasibility studies, which allows investigators to come with a proposal to the FDA for a new device and for an IDE, investigational device exemption, when you haven't finished everything. So you're not going to once this is approved, I'm going to market this. This is a novel product that's in development, and it's going to be iterative. So we're going to do a small number of patients and then reiterate, regroup, maybe change the design, do another study with a small number of patients. And with the idea that the investigator and the FDA can be very collaborative over this. And our experience has been extremely positive in terms of this collaboration. And I've already mentioned the translational integrator function. The delayers, I think, as I mentioned, funding. If the funding level goes down, then that slows things down. The difficulties in managing those academic, commercial, industry relationships. And then some of the approval processes within the Department of Defense, especially when you get close to human studies, can be extremely slow. But they're essential. You can't move on. They tend to be duplicative with your own institutional processes. So once something's gone through your IRB, and maybe even the FDA, it also has to go through HARPO, which can sometimes be just a very, very slow process. And I can give an example during the discussion time. So I'll stop there and just say that from the investigator's perspective, there are pluses and minuses, but the overwhelming message is that this is a very positive funding source, and it's really a very exciting agency to work with, especially with the end result being trying to restore our wounded young warriors back to the best quality of life possible. So I'll stop there and let you introduce the next speaker. Thanks.